मेडिसिन लेके आपका बीपी कंट्रोल है तो आप कर सकते हैं, बट विंटर्स में सब कर सकते हैं 5 to 10 राउंड्स। देखिए आपकी बॉडी के अंदर कितने चेंजेस आने स्टार्ट होते हैं। वेरी गुड फॉर द कोलेस्ट्रॉल पर्सन। तो सूर्य भेदी यानी कि सूर्य को भेदना, तो गर्मियों में हम लोग किससे करते हैं, ठंडी नाड़ी से स्टार्ट करते हैं, और सर्दियों में हम लोग किससे करते हैं, गर्म नाड़ी से। तो दैट इज कॉल्ड एज सूर्य भेदी प्राणायाम। अब हम दूसरा प्राणायाम जो हम सीखेंगे, 3 प्राणायाम सीखेंगे। दूसरा प्राणायाम जो है हमारा भस्त्रिका प्राणायाम। भस्त्रिका मतलब क्या होता है, लोहार की धोपनी। पुराने जमाने में जब आंटियां खाना बनाती थी तो उनके पास में एक ऐसी स्टील की पाइप होती थी और वो लोग क्या करते थे, इस तरीके से फूंक मारती थी, है ना। तो ये हमें क्या करना है, ऐसे धोखने की तरह अपने लंग्स को पंप करना है। हार्ट की पंपिंग उससे बढ़ेगी, एक्स्ट्रा कोलेस्ट्रॉल जो होता है वो खत्म होता है। और सबसे इंपॉर्टेंट जो इस भस्त्रिका प्राणायाम का बेनिफिट होता है, देखिए अभी आप ये कॉन्फ्रेंस अटेंड करने वाले हैं, हो सकता है कोई लेक्चर बहुत अच्छा हो, कोई लेक्चर हम चाह के भी हम सुनना चाहते हैं, बट अगर आफ्टर लंच के बाद अगर सेशन होता है तो इट्स वेरी वेरी कॉमन कि हमारा जितना भी ब्लड होता है वो कहाँ चला जाता है, खाने को डाइजेस्ट करने में चला जाता है। तो हमारी बॉडी के अंदर किसकी डेफिशिएंसी हो जाती है, ऑक्सीजन की डेफिशिएंसी होती है। एक प्रोसेस होता है जिसके थ्रू हम अपनी बॉडी की थकान को खत्म करते हैं, जिसको हम बोलते हैं यॉर्निंग। आप में से बहुत बार कई बार ऐसी मीटिंग के अंदर भी सिचुएशन आ जाती है कि हम नहीं सोना चाहते हैं, नहीं यॉर्निंग करना चाहते हैं, बट हमारा कंट्रोल ही नहीं होता है। हम क्या करते हैं, यॉर्निंग के थ्रू अपनी बॉडी के अंदर जो एक्स्ट्रा CO2 कलेक्ट हो जाती है हम लोग उसको बाहर निकालते हैं। अब ऐसी कंडीशन हो, आपके सामने आपके बॉस बैठे हों और आपको बिल्कुल यॉर्निंग वाली कंडीशन आ रही है, तब आपको क्या करना है, भस्त्रिका प्राणायाम करना है। अब भस्त्रिका प्राणायाम जो एक्चुअल जो प्रोसेस होता है वो हम किसी के सामने तो नहीं कर सकते हैं, बट उसको कैसे फॉर्म में हमको करना है मैं आपको वो भी बताऊंगी। भस्त्रिका में क्या होता है, फोर्सफुल एक्सलेशन, फोर्सफुल इनहेलेशन, वेरी गुड फॉर लो बीपी। जब भी आपको शाम के टाइम पे लेथार्जीनेस लगे, आपकी बॉडी के अंदर आपको एनर्जी की डेफिशिएंसी लगे, तो आपको क्या करना है, इमीडिएटली, मतलब बोलते हैं ना इंस्टेंट पावर अगर आपको चाहिए, जैसे ग्लूकोज हमें देता है, उसी तरीके से भस्त्रिका आपकी बॉडी के अंदर इंस्टेंट पावर देने का काम करता है। तो क्या करना है, आपको क्या करना है, दोनों हाथों से मुट्ठी बनानी है, अंगूठा हमेशा अंदर की तरफ, फिंगर्स जो है वो आपकी कैसी रहेंगी, बाहर की तरफ, और फोर्सफुली पूरा सांस भरना है, और फोर्सफुली आपको क्या करना है, एक्सहेलेशन करना है। जितना फोर्सफुली कर सकते हैं, अपने लंग्स को पूरा ओपन, क्लोज, कॉन्ट्रैक्ट, रिलैक्स, अपनी बॉडी के अंदर मैक्सिमम ऑक्सीजन को आने दीजिए, और CO2 को आप लोग क्या करिए, बाहर करना स्टार्ट करिए। अब आप कहीं बैठे हैं, मीटिंग के अंदर बैठे हैं, आप अपने हाथों को तो मूव नहीं कर सकते हैं, बट अगर आप कॉलेज के स्टूडेंट्स को ये प्रोसेस करा सकते हैं, अगर आपकी आफ्टर लंच कोई क्लास है, आप क्या कर सकते हैं, अगर आपकी सिचुएशन किसी के सामने अगर ऐसी हो जाती है, तब आपको क्या करना है, 5 टाइम्स खाने खाने के बाद में आपको कोई मीटिंग अटेंड करनी है, कोई क्लास अटेंड करनी है, कोई सेशन अटेंड करना है, फोर्सफुल इनहेलेशन, फोर्सफुल एक्सहेलेशन, इसको करिए और देखिए आपको कैसे पावर मिलती है। तो भस्त्रिका इज वेरी वेरी पावरफुल प्राणायाम जो आपको इंस्टेंट एनर्जी देता है। जिनको भी लेडीज को जनरली कॉमन प्रॉब्लम रहती है लोअर बीपी के लिए, तो उनके लिए तो बिल्कुल बेस्ट प्राणायाम है टू गेट इंस्टेंट एनर्जी। लास्ट प्राणायाम हम जो करेंगे उसके साथ में हम उसके बाद में अपनी आंखों को नहीं खोलेंगे, 5 मिनट का मेडिटेशन भी हम उसके बाद में करेंगे। लास्ट प्राणायाम जो मैंने आपको कॉमन प्रॉब्लम बताई है, स्ट्रेस की, एंजाइटी की, डिप्रेशन की, उसके साथ में आज का एक कॉमन प्रॉब्लम हो गई दैट इज कॉल्ड एज माइग्रेन हेडेक। बहुत ज्यादा है लोगों के अंदर, क्योंकि वो सोचने की जो प्रॉब्लम बढ़ चुकी है, हम हर चीज में ओवर थिंकिंग जो हमारी प्रॉब्लम हो गई है, तो कईयों का तो पूरा ब्रेन पेन करता है, कईयों का एक साइड राइट साइड या लेफ्ट साइड ब्रेन पेन करता है, तो उसके लिए बहुत अच्छा है। एंड फिफ्थ जो कॉमन प्रॉब्लम है, इनसोम्निया, यानी कि नींद नहीं आती है। एक होता है कि हम 10 बजे हम बेड पे गए ना, आधे घंटे में हम सो गए, विद इन 10 मिनट्स हम सो गए, और एक होता है 1-2 घंटे के बाद, 3 घंटे बाद में ओवर थिंकिंग के बाद में हमको नींद आ रही है, तो दैट इज कॉल्ड एज इनसोम्निया प्रॉब्लम, कि रात में हम जब सोते हैं तो 2-3 घंटे तक या आने के बाद में खुल जाती है। तो भ्रामरी सबके लिए बेनिफिशियल है। भ्रामरी को आप सोते टाइम पे भी कर सकते हैं, अपने बेड पे लेट के भी कर सकते हैं। भ्रामरी यानी कि भंवरे की गुंजन, भ्रामरी यानी कि आपका हमिंग साउंड आपको प्रोड्यूस करना है। देखिए इस प्राणायाम के बाद में हम अपने हाथों को नीचे कर लेंगे और अपनी आंखों को बिल्कुल बिल्कुल बंद रखेंगे। आपको ये प्राणायाम कैसे करना है, दोनों अंगूठे आपके कान पे आएंगे और फिंगर्स आपकी आईज पे आएंगे। एक बार आप देखेंगे इसमें एल्बो कभी भी नीचे नहीं रहती है, हाथों को बिल्कुल आपको ऊपर की तरफ रखना है। बिल्कुल हाथ आपके ऊपर रहेंगे, दोनों अंगूठे कान पे, फिंगर्स ऑन योर आईज। एक बार आप पहले देखिए, पूरा सांस भरनी है और म का साउंड प्रोड्यूस करना है। हम्म। इस तरीके से अभ्यास करेंगे, जितना लंबा म ले सकते हैं लेने की कोशिश करिए। तो सांस भरते हुए 3 बार म का उच्चारण। हम्म। 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 आंखें बंद रहेंगी, ध्यान सांसों पे। टेक अ डीप इनहेल एंड एक्सहेल। सांस भरिए, सांस छोड़िए, आंखें बिल्कुल बंद रहेंगी। लंबी गहरी सांस लेंगे, लंबी गहरी सांस छोड़ेंगे। कमर सीधी, गर्दन सीधी, कंधे ढीले, शरीर एकदम ढीला, बट कमर हमारी सीधी। 10 बार धीरे धीरे अपने ध्यान को, मन को, चित्त को अपनी सांसों पर लेकर आएंगे। महसूस करेंगे अपनी हर एक सांस को, कैसे आ रही है, कैसे जा रही है। हर एक सांस पे अपने ध्यान को, मन को, चित्त को लेकर जाएंगे और काउंट करेंगे अपनी ब्रीदिंग को फ्रॉम 10 to 1। 10 सांस लेते हुए, 10 सांस छोड़ते हुए। स्टार्ट काउंटिंग ब्रीदिंग इन योर माइंड फ्रॉम 10 to 1। महसूस करेंगे आपकी हर एक सांस एकदम सामान्य हो चुकी है, एकदम रिलैक्स। धीरे धीरे अपने ध्यान को, मन को, चित्त को अपने आज्ञा चक्र पर लेकर जाएंगे, यानी कि अपने दोनों आंखों के बीच में। इसे महसूस करेंगे अपने दोनों आंखों के बीच में, अपने ध्यान को, अपने मन को, अपने चित्त को। बस अब आप हैं और आपका ध्यान, मन, चित्त आपके आज्ञा चक्र पे। आपके आज्ञा चक्र पे आपको एक ज्योत, एक प्रकाश, एक लाइट दिखाई दे रही है। उस लाइट को, उस प्रकाश को देखने की कोशिश करेंगे। बस अपने दोनों आंखों के बीच, अपने त्रिनेत्र पे एक ज्योत दिखाई दे रही। ये ज्योत अब धीरे धीरे और तेज हो रही है। इसकी रेस चारों तरफ फैल रही है। इस ज्योत के साथ में हमारा तेज, हमारा प्रकाश, हमारी रोशनी बढ़ती जा रही है। हमें भी इस ज्योत की तरह अपनी रोशनी को चारों तरफ फैलाना है। अपने प्रकाश को चारों तरफ फैलाना है। धीरे धीरे ये ज्योत अब हमें ओम में दिखाई दे रही है। इसे महसूस करेंगे, महसूस करेंगे। ओम में भी, ओम के बीच में आपको वही ज्योत, वही तेज, वही प्रकाश दिखाई दे रहा है। आपकी आंखों के सामने एक बहुत बड़ा चमकता हुआ ओम आपको दिखाई दे रहा है। उसे देखने की कोशिश करेंगे और धीरे धीरे अपने ध्यान को, मन को, चित्त को ओम की तरफ ले जाते हुए 3 बार ओम का उच्चारण पूरा सांस भरते हुए अच्छे से करेंगे। ओम, ओम, ओम। आंखें बंद ही रहेंगी। आपका मन, चित्त एकदम शांत। दोनों हाथों को अच्छे से रब करेंगे। आंखों पे रखेंगे। अपने हाथों की पूरी ऊर्जा को अपने हाथों पे और अपने शरीर में लेकर जाएंगे। धीरे धीरे ईश्वर को धन्यवाद देंगे और एकदम शांत, एकदम रिलैक्स, एकदम शांत, एकदम रिलैक्स इस ऊर्जा को अपने साथ में रखेंगे। खुश रहेंगे, स्वस्थ रहेंगे। थैंक यू सो मच सर। थैंक यू पंकज सर। थैंक यू IIT जोधपुर फॉर इनवाइटिंग मी। अगर आपके कोई भी क्वेश्चंस हैं तो आप पूछ सकते हैं।
Thank you so much, ma'am. It was really a very interactive and insightful session. And I personally learned a lot of things out of it. So let me check if there are any question
Sure.
from panelists first, then we can go to audience attendees. Is there any question? Please feel free to ask from panelists first. Okay, so seems no questions. Let me just please wait. Let me check if there are any questions from attendees. Attendees, please raise your hand if you have any questions, or you can type in the chat box.
I think everybody has become very peaceful after the session.
Yeah.
Thank you.
Okay, so if there are no any questions, maybe I can ask one question. It's not related to yoga, but this is more little bit of scientific question. So, are you aware of any tools or any way by which we can say, okay, the person is doing the yoga in the right way, the asanas in the right way or not? Is there any kind of work that has been done in this direction?
Yes, sir, definitely. We have a tool for this. I will send you on the email so you can correct it whether you are doing the right way or not.
Oh, okay, that's great. That will be really helpful because most of the people, when they
Yeah,
start
looking for the paper,
making,
yes,
they
sir.
don't know whether they are doing it in the right way or
No,
not.
many tools has developed for this, sir.
Okay, great. Okay,
Thank you,
thank you
sir.
once again, ma'am. And with this, I would like to thank, and
Om
we will
Shanti,
move to
sir.
the next speaker. So, Monica, you can take over now.
Can you also share, give the share permission?
Yeah, and also, Ashish, please assign share permission for Professor Mitali.
Shall I start, sir?
Yes, you can, Monica, please.
Good morning, everyone. It is a great pleasure for me to introduce our speaker for today's talk, Dr. Mitali Mukherjee, ma'am. Dr. Mitali Mukherjee is a professor and head of the Department of Bioscience and Bioengineering at IIT Jodhpur. She was formerly a chief scientist at the CSIR Institute of Genomics and Integrative Biology and was involved in setting up key genomic initiative in the area of rare genetic disorders, population genomics, functional genomics of repetitive sequences, and genomics. She has a PhD in molecular genetics from the Indian Institute of Science, Bangalore. She has been the convener of the Indian Genome Variation Consortium project, which provided the first comprehensive genetic landscape of the Indian population. She has been involved in co-developing a new area of research, Genomics, which aims to integrate Ayurveda with modern medicine. She has received a number of national awards, the CSIR Young Scientist Award, DBT Young Women Scientist Award, and Shanti Swarup Bhatnagar Award, and many more. She is also the board member of International Consortium on Human Phenomics. Without any further delay, please join me in welcoming Dr. Mitali Mukherjee, ma'am, to address the session on phenomics. Thank you so much. Over to you, ma'am.
Thank you so much for giving an opportunity to speak over here. So there is a title called Phenomics over there, but I felt that it might be more pertinent to kind of give a flavor of genomics and what all kinds of questions you can ask through it. And some of it would be my journey into this area and how do we go ahead from there. Okay, so so once again, welcome to IT. And the way I've designed the talk is how we can connect different sciences through the interface of genomics. That's the area which I've been working on for the last 2 decades. So, so one of the things which I also mentioned in the beginning of this day is that we are very curious to understand design principles from the biological intelligence of natural systems. And I talked about how this has inspired a large number of engineering marvels and how people are also trying to understand different aspects of various kinds of animals in this planet, as well as understanding ourselves. So, so the very interesting thing is that all life forms on Earth are mainly made out of these four chemical letters, except, of course, many RNA viruses, most prominently the viruses of the kind of COVID, which we are being we've been becoming very familiar in the last 2 years. We've become very familiar with that, and this allows us to, even if there are all made from the four chemical letters, the combinatorial properties of these letters have actually given rise to such diversity. And had it not been the seminal elucidation of the structure of DNA by Watson and Crick, we would not have had such detailed understanding about how these form the fundamental units of genetic material. Now, even though they are all made out of the same chemical letters, which form what we call as the book of life, these letters make different kinds of sequences which we can so the DNA gets threaded into these meaningful words which we call as codons, and threading of these codons makes these uh, meaning which are like the sentences, or these are called as genes, which have punctuations as in a sentence we read. And if these sentences are threaded together into chapter, which these would be the chromosomes. So from the DNA, you get meaning into genes, and those genes are organized into chromosomes, and the entire set of chromosomes is what is called as the book of life, right? So these DNA are hardwired, right? You have this sequence read all over in every cell, but then how do you get the meaning out of it is through this process of transcription. So the DNA gets into some kind of a meaning by through a messenger, which reads these nucleotides and converts it again into a thread, which are read in threes, and these are what are called as the codons. And what these are converted is through a process of translation into proteins, which we all know about. So these are read in threes to incorporate one amino acid, and this threading of amino acid together makes what is called as the protein. So you can very well imagine if there is a change in the DNA, then that might lead to altered codon, and that would lead to an alteration in amino acid, and then you might have a changed protein. So such kind of changes are very common. These are when the DNA is making copies of itself. It can change a little bit. Some of these changes might be of different kinds. So you can have a change where there could be a deletion, there could be a substitution, there could be an insertion, there could be inversion, or there could be fusion. And so you might start with the word like color, and through these small changes, you might actually end up to being a cool look, right? So such kinds of changes are what are called as variations. And if the changes are very infrequent and very rare, occur in a very less number of people. So sometimes, because these some of these variations might not have meaning, and might not have meaning in terms of the protein is might make a non-functional protein. So these would be called as mutations. Mostly they are very rare, and there could be some changes which could have still some meaning. For example, color and color would be spelled differently in different parts of the world, so it could have a meaning. So these would be what would be called as common variations. So one of the kind of variations which most of us are aware of is this single change, where one amino acid becomes another amino acid in the beta globin gene, which gives this different shape to our red blood cells, and the red blood cells become like the sickle shape. And this is a kind of a condition which is called a sickle cell anemia. So your oxygen carrying capacity is compromised just by a single change. So these are the kind of changes which can happen in a genome. And long back in the 1980s, then it was thought that if you are able to read all the nucleotide in the book of life, in the sequence, then one can actually see how different we are from each other in disease condition, or some other kinds of phenotypic changes which we makes us all different. And then we would know the book of life, and that could be a reference book for all of us. So this was what was called as the Human Genome Project. And the Human Genome Project took 20 years, but after that, there had been a lot of technology advancement, as a result of which not only human, but lots of other genomes have got sequenced, and even many human genomes now can be sequenced in 3 days' time because of many technology advancements which happened during this period. So the idea was, as I mentioned in the first part, that if one were to know this DNA sequence in a reference, idea was that if you would be able to compare and understand which are the changes which are not good in prenatal stages or in fetal needs and intervention, so you could actually have these things understood right from the womb to the cause of death. So this all has been made possible because of so many advancements. So these were the first generation sequencers which were involved, which were used extensively for human genome sequencing. But with rapid advancements, there have been other sequencers, the next generation sequencers. I'm sure you'll get to know more about it as the workshop proceeds. And now we actually have something which can be held in the palm top and could be equally powerful in reading the genome. So these are called as the nanopore, and this has made sequencing possible in even very remote locations, and you don't need to depend on these very expensive high throughput machines anymore. Because of these kinds of advancements, people have become very ambitious, and then there's a big project called the Earth Biogenome Project, which actually aims to sequence all kinds of flora, fauna, birds, country-wise counting or country-wise understanding what are the various designs of different kinds of living organisms on Earth. So this is and why these so are these sequences revealing? The answer is yes. One can ask many interesting questions. For example, honeybee, all the bees in the hive have the same genome, but some become queen, some become drone, some become workers. And what is it within them? Why do they behave very differently even from the same genome? Or why is there so much water in watermelon? So people have found that the vascular system, which allows them to kind of accumulate so much water, is differently. Those sequences have to evolve differently over here. People also ask, what is the smallest genome? And then we could have a kind of in an organism or in a living form. And for example, this one is a carnivorous plant. It has got a very short genome. It has got rid of all those things which it doesn't need, because it can get all its nitrogen source by feeding on insects. So its genome is very small. Or there are certain kinds of bacteria which can withstand huge amount of radiation. And that's because their genomes reveal that they have very effective repair proteins, which allows them not to disintegrate in such UV conditions. So not only we are able to know each of these genome story, but because of this very interesting attribute that they are all of the same made of same nucleotides, nature have also used them as Lego pieces, and sometimes there are shared nucleotide sequences which have the same functions in different genomes. So people are also finding these things from studying small organisms, and they are finding those very genes, if there are changes, also lead to many defects even in the humans which share these sequences. So for example, studying bats, you can know why you have multiple limbs, or the reason for pigmentation abnormalities, or why elephants do not have cancer. And all these things allow us to understand what happens if there is a disease, and what happens if there is how do you know who are the people who might not get the disease by looking at these sequences. So So there has been a information also of very, very interesting kinds based on the behavior of animals and looking at comparing them with the humans. For example, this is a very, very interesting study about hibernating animals. Before they go for hibernation, they eat quite a lot. They are called as polyphagic. And because their metabolism has to be reduced when they are in hibernation. And it turns out that those very genes are actually involved in the obesity, contributing to obesity, because the habit of eating lot and metabolism low is also a shared feature. And people looked at the genome or their expression in hibernating animals, and they found that they share sequences to those stretches of the human DNA which are involved uh, uh, in contribution to obesity. So this is what is the phylogenomics aspect, and many, many more such kind of answers come. We've also studied how loss of body hair would need a very, very good barrier function by this keratinization. And we studied various animals which have different relations to water, and we could find that the genes which are involved in keratinization could have contributed majorly to protecting our skin from not kind of meaning being an evolutionary adaptation when we lost our hair during evolution. Genome sequencing, all of you know now that this has been a major kind of technological help during the COVID times, because this allowed us in a very short span of time to track the distribution and spread of SARS-CoV-2. This is a site which is being maintained by CCMB for Indian genomes, but what it allows you is, as people are sequencing, and if they start depositing the sequence in the real time, one can know what kind of viruses are prevalent at that particular time, so that one can actually use it to track them and try to find which part of the country has what genomes at what particular time. So these have been really, really seminal in today's time. Now, coming to this was all about the genome, but as I mentioned that the Human Genome Project was all to understand about whether we can get to a reference. But what came out was that human is not only human because of their genome. There are lots of variations in the human genome. These could be single nucleotide changes, these could be short indels, these could be bigger changes. And all of these contribute to not having a reference genome, but there are multiple references. And then there was another area which came up because of the ability to sequence. So people started sequencing microbes within the human, and we could find that the human has more than 10 times the number of cells than in the what we have as human cells. And these would contribute to a large number of genes which come from different kinds of bacteria. And so human is not only the expression of the genome from the human cells, but also expression of the genomes from the microbiome which resides in us. And then all these also are dynamic because they could be shaped by environment, by the way we have migrated to different parts of the world, how we have admixed, or the kind of cultural practices we have. So today's date, it's become a challenge to just study the genome by its sequence, but also to contextualize it with respect to its expression, be it microbes, be it RNA. So there are multi-omic methods now needed to be able to understand what would be even a normal human genome. So we still are very little far from describing from the genome what is health or who is healthy. Because today's day, the definition of health is always decided in the context of any disease. You don't have asthma, you are healthy. You don't have diabetes, you are healthy. But how healthy you are becomes a challenge. Nevertheless, lots of questions are getting answered by looking at these variations. So one can compare variations amongst different populations, and one can individuals, and one can look at sharing of variations between individuals to be able to know whether they are likely to have the same kind of background. And that's how lots of theories about humans, human migrations, are getting validated by such kind of genomic technologies. For example, it was known that humans would have evolved in Africa 100,000 years back, and they would have moved to different parts of the world through various waves of migration. Now one can actually study the genomes of people who are residing in the different parts of the world and try to compare their genetic information, and then one can start to cluster them using the methods of data sciences, and try to group them together. So this is one such study which was done on nearly 72 populations from different parts of the world, from Africa to Middle East, to Europe, to South Central Asia, to East Asia, Oceania, and America. So people had been sampled in different parts of the world, and you could see that the colors are similar. That means to say that if you look at different parts in the human genome and try to see how they are related to each other, you end up clustering populations in Africa together, and the Middle East are a little different from them because they have a little different color, but they have admixture with the European population. Then Central South Asia is a little different from Europe, and East Asia even different. So as the humans have moved, their genomes have changed, and as a result of which today's day, if you look at the genomes of human in Africa versus genomes of human in America, they would be very, very distinct, and one can use this kind of an information to trace ancestry. But what would be very interesting is that supposing there is a mutation in a person in a particular background, then you can also start to trace it in other population where the mutations would have come by overlaying this kind of a pattern of background relation. So such kind of studies are very revealing. For example, if you look at this map of the world, this is a map of exposure to UV. So this part, the tropical region, as all of us are aware, is very, very intense in the UV exposure. And what it does is, if you have a UV exposure, you tend to see people with a darker color of skin. And that's because then you can prevent the folate in our body from getting degraded. But what happens is UV is also essential for vitamin D production, which is also important in a large number of processes in the body. So as a result of which the darker color skin might not prevent much UV getting in. So you might be a little low in vitamin D. Whereas in the lighter skin, you could also have the higher UV exposure, but then that might be needed in parts of the world where there is a very low level of UV exposure, meaning lower periods of the time, and as a result of which the vitamin D production might get affected. And that could have led to having evolution of lighter skin or darker skin, depending on the kind of environment you reside in. Now, it turns out that this is what is the skin pigmentation pattern. People who tend to stay very close to regions of high UV exposure have a darker skin, whereas places where the exposure is lesser, people have lighter skin. And if what happens to the genetic makeup? So it turns out that there are certain genes which are involved in conferring differences in skin pigmentation. They differ between populations of Africa and populations in Europe. And such kind of information then becomes very relevant in diseases, or in even in animals which have differences in color. People find that such kind of genes are also involved in differences, conferring differences to such kind of skin differences. And these regions which differ as a consequence of the climate also give important clues to understanding the basis of diseases which are involved in things like depigmentation or skin cancer, melanoma. It can also give you other kinds of information. For example, there are foods which are bitter, and there are people who don't take bitter food, and so they would be avoiding cruciferous things, or alcohol or caffeine. But so these are also there in our genetic makeup, makes us perceive a food differently, as a result of which there are some super tasters to whom very low amount of bitterness also is perceived as very highly bitter, and these are because of variations in the genes which allow us to perceive bitterness. So these are the kind of convention we follow to say there is a variation in a bitter taste receptor gene. And if people have such kind of variation, then they would tend to avoid food of this kind, and they would tend to like food of this kind, and which might give us a risk for high BMI, obesity, etc. So having a knowledge of such kind of variations, if you have, then you might also become mindful eaters, because then you would need to, because you need to have such kind of food and maybe avoid this kind of food for a better quality of life. So this is what is the area of precision nutrition, and this is what is also coming up very much. So in the Indian scenario, we also wanted to understand how the Indian populations are organized, because Indian populations are very, very diverse. There have been large number of movements within the population, lots of invasions from different parts of the world in different time periods. And because of the kind of way we are socially, culturally organized, there are thousands of communities, differences in language. People in different parts of the world speak different languages, or they have different kinds of morphological features. So if you say what would be a reference Indian genome, or can you look at the DNA and say this person might be coming from this part of the world, we needed to build a genetic map of India. So this was one of the projects which I was involved in. It was from 2002 to 2007. It was called as the Indian Genome Variation Consortia, and we wanted to know how, as Indians, we are related at the genetic level. This was a major project with more than 200 people participating, and this was done at CSIR level. But the idea was that at that time, lots of other people were generating data on different parts of the Indian population. There was nothing which was available from India, and we decided to identify whom would we call as reference population. Are people who reside in the same linguistic map, are they more related to each other, or is there something more beyond that? So this is a genetic map of India. Each of these pie chart represents a population which we studied in different parts of the country. And as you can see, the bottom color is a linguistic map. In the same map where people all speak Indo-European language, there are different color pie charts, which says that there are people of different genetic background. And so we wanted to understand the meaning of this as to who are more related to whom, and how we are related to the world population. So it turns out that if people are speaking Indo-European language and come from the large population and residing in the north, they are more related to each other. Populations which reside in the Himalayan belt, but they could be speaking Tibeto-Burman or Indo-European or Austroasiatic language, they are more related to each other. People in this belt who are mostly of tribal origin and can speak Austroasiatic or Dravidian language are more related to each other. And in the southern part of India, there are a set of populations who are more related to each other, but there have been also more very, very extreme north and extreme south have more relations, but it was known from archaeological evidence that initially these people might have been resident here, and as there have been invasions, they were pushed down. and there are certain populations which are more similar to the North Indians here. So this is the kind of map we built, but then this was the static map. And then what we could do is we could actually overlay different kinds of things and start asking a question that, okay, people are related to each other, but what happens if they reside in particular region? For example, if that's very malaria endemic. It turns out that there are certain genes which are involved in malaria resistance pathway differ in malaria endemic or non-endemic region. So then this gives us a hint that if people are residing here and are braving malaria, then they could have a different genetic makeup which protects them from malaria. So you could use these kinds of information to then study whether if people of this background are residing in non-malaria region, would they be more protected from malaria? So many of our colleagues have taken such kind of insights from the map to try to understand, let us say, protection from diabetes, protection from HIV, etc., whether we could take kind of insights from here. Like I work with Dr. Saman Habib in CDRI, and we could actually find that there is a gene which differs in its distribution, in a variation in this gene differs in distribution between malaria endemic and non-endemic region. And this distribution was similar to the meaning you could see that a particular kind of variation is more present in endemic region. So we went about taking people who had got malaria, falciparum malaria, from this region and from the other region, and we found that having this kind of a makeup protects people from malaria, and that's how we use these kinds of baseline information. So, but this differs in endemic and non-endemic regions. So if you have even one of this variation here, then they would be very, very susceptible to malaria. So I'm just trying to show that how you can use variation data in asking different kinds of questions. There was a question as to how we are related with our neighbors, the South Asian neighbors. And this was a very interesting study we conducted way back in 2009 with different parts of the Asian populations. There were 72 populations from 10 different countries, Singapore, China, Taiwan, Japan, Philippines, Indonesia, Malaysia. And we asked a question how we are all related to each other. And there was a very, very interesting finding that we find that from the Europe, from Africa to Europe, when people migrated to different parts of Asia, there has been one major kind of movement first into India and then into different parts of the world. So India seems to be a melting pot for human diversity, which has been told many years. This is something which came out as an evidence in this kind of a work. There are other interesting studies one can do. What happens to people if they move from non-native to native environment? So do their genetic makeup of habituation in one particular environment, does it allow them to kind of adapt to a different environment? We asked one such question from a particular set of people who are known to have been coming from Africa with the slaves, with the Portuguese sailors, and they have come to reside in Gujarat, and they are called as Siddhis. And they nearly look like Africans, but they speak Hindi. They've been converted they are mostly a Muslim community. And when we look at their genomes, we find that they are very different from the Indian population. Now, we wanted to study them even a little more because there was an interesting story linked with them, that these people come from Africa, from Ethiopia. That's how we could trace using the DNA. And they have come to reside in a very high salt region. Africans, when they are in Africa, they sweat quite a lot and they get to kind of lose salt, as a result of which they have a variation which allows them to retain salt. Now, it's known that when Africans move to Europe, America, they are more hypertensive because they do not sweat so much, and they have a copy of meaning those variations which allow them to retain salt. So we were interested to know whether these people, if they have come to Gujarat, where there is a lot of salt, what happens to their genome? So we looked at their genome, and it turns out we can actually trace in each of their genomes whether this part is coming from Africa or this part is coming from India, and this is by something called admixture mapping. You look at the regions of the genome which are more similar to Africa and regions of the genome which are more similar to India. And since this is a very, very recent population, only dating to nearly 200 years, the genome would not have changed in many places. So long segment of DNA can be traced to some kind of ancestry. And what we found was that the regions which came from Africa had, or from India rather, had genes which are linked to many processes which are relevant in salt adaptation. So this seems to be a very, very extreme case of selection, where if people who are not habituated to salt environment come to India and admix with local population, mostly they might acquire certain genetic regions which are more relevant to adaptation in such kind of environment. So this kind of a way where we have used a huge amount of such kind of natural variations from the Indian genome variation data to understand how we are related within a population, how we are related to the world population, how who have migrated and whether the genetic footprints tell about migration. And from the migration, could we also get idea about how diseases spread across different parts of the country? And then could we use them for other kinds of studies to identify which are the populations which could be at risk of certain kinds of things? And how do you identify from here who are likely to adapt to different kinds of environmental stresses? And also whether you can actually get even more newer insights. So many, many papers have come out where Indian genome variation is actually an author. And this is really shows that how, if you actually have a baseline information, then how you could use it for different kinds of studies. For example, now we are in the desert, and I got interested in studying people in the desert. So one of the interesting thing about desert is people have a lot of consumption of chilies in hot climate, because that allows you to cool your body by inducing sweat formation. It is also a stimulant, and it also has antimicrobial properties for preventing food spoilage. It turns out that there are variations which allow you to perceive pungency in a food, and such kind of variations differ in populations which are habituated to spicy food, because if your genetic makeup did not change, then you would be averse to certain food which are very important for that ecoregion. So these are now you can see cultural conditions or cultural adaptations, or are also kind of provide certain meaning. Certain genetic makeup could also allow you to identify if there are variations linked to such kind of cultural adaptation. So understanding genomes can allow you to identify signatures which are relevant in evolution or selection. And it can also give rise to lots of diversity, both at the organism and organismal level. And looking at the patterns in the genome, then you can actually understand unique biology, phenotypes, and diseases, which could tell a lot about history, ancestry, culture, ecosystem dynamics. Lots of kinds of things can get to know from such kind of studies. Just the last few. Pankaj, I have some time.
Yes, ma'am, you have sufficient time.
Yeah,
10,
yeah.
15 minutes you can take this.
Yeah. So now having such kind of a baseline, how is it relevant for today's solutions, right? So this is much become more relevant in the COVID pandemic time, because each one of us in different parts of the world has been impacted by the same virus, whose origins have been traced all to one common source, and it's evolving rapidly. But what is getting very evident is it's impacting all populations of the world, but everybody is not getting affected to the same extent. There are certain populations in which there has been very, very high prevalence there, whereas there are certain populations which might be relatively protected. So people have started thinking that there is a role of the environment along with the genetics. And also within a population, even if in a family, many people are getting affected, one doesn't know why there is a different outcome. And this is very important for everybody to know in a family, as we would have all experienced that if we were to know who is likely to have what kind of a different outcome, could we actually manage ourselves much better, or could we have a better prevention in place? The second thing is that such kind of thing also requires rapid solutions to identifying vaccine candidates, drugs which are already in the market for repurposing. So this COVID pandemic has actually, for the first time, made lots of people aware across the world about the genomics, and also about appreciating that genomics itself is not enough. You need to understand population differences, and you need to understand inter-individual differences. This is where I just kind of want to come to this Ayurveda system, which is our ancient system of medicine. And this is one of the most precise system of medicine which is documented. And this has also been acknowledged in the recent times that in Ayurveda, people are identified on the basis of these constitution types, based on which the treatments are tailored. So if you look at this shloka from the Charak Sutrasthan, it's called, you will find that there is this definition which English translation says, aim of Ayurveda is how you maintain health in healthy and alleviation of disorder in diseased. So we've been studying for the last 2 decades as to how they knew, and can we actually get an evidence in today's time so that its acceptability or its understanding for other discoveries become much more possible. So what is this constitution? So in Ayurveda, this Prakriti is assessed by looking at a person from various windows, their anatomy, their body build, their body frame, how is their physiology wired in terms of their digestive capacity, in terms of their appetite, many such features in terms of how they walk, how they talk, how they move their body parts when they are talking, or also their psychological attributes. So there are nearly 200 kind of features in which people are examined, and you could make out a person's Prakriti from there. What is very interesting is that any population, people are kind of classified into various kinds of Prakriti types, broadly, which can be binned into 7 groups, which are called as Vat, Pitta, Kapha. There are combinatorials, and then there are people who are also all the 3 together. What's interesting is Prakriti of a person doesn't change when they are born. If they are of Vat type, then they remain Vat, but we clearly show differences, so that how we age at different times of our life is also governed by what our Prakriti is. So Prakriti tells about what is the state of health of a person, how the person is likely to respond to certain kinds of environmental trigger, what are the kind of disease trajectory they can take, and how one individualizes them. So this seems to be something which also the precision medicine wants to address from womb to tomb. And how this comes about is they have certain conceptual framework where they talk about people in the context of having these uh, different proportions of something which is called as three dosha. So three dosha are 3 physiological components. One is called as the pith, the other is called as the vat, and third one is called as a kapha. And people would have varying amount of vat, pitta, and kapha in their system. So if individuals have more vat in the system, they would be called as vat type or pith type or kapha type, depending on which dosha is more prominent. But what is very interesting is diseases are also described as a perturbation of this dosha from their homeostatic state. So, for example, if more Vata increases in a person, then this person is likely to have a disease, but this would be more from perturbation of Vata dosha. Pith people can also have Vata dosha perturbation, but then for the Pith person, this person moving into even this direction, this much direction will be a disease, but for Vata person, this is not a disease state. So one looks at a person's basic dosha constitution, and from there they try to assess what is the perturbation and in which direction, and that is what is called as a vikriti. And all the medicines are basically mapped onto which getting which dosha into balance, and this is how Ayurveda practices looking at person from their dosha perspective to what diseases they have and how they would be getting back to homeostatic state. So everything is all about dosha. If you look at the causes of the disease, if you look at the phenotypes of a disease, and if you want to evolve a particular therapy. So this whole interconnection understanding of causes and the features and the therapy is what is called as Tri Sutra, and this forms the basis of how Ayurveda describes personalization. Now, what is very interesting is that it also talks about what is Vata. With this, these would sound like abstract components, but there is a vivid description of all of them as to what governs what kind of properties in the body. So everything to do with shape, cell division, proliferation, signaling, movement. These seem to be like biochemistry terms, but there are Sanskrit shlokas which are, this is a literal translation from the shlokas of such kind, which will tell what are the functioning of Vata, Pitta, and Kapha. Now, today's date, if you look at biochemical pathways, then you also have these kinds of terminologies there: signaling, movement, transport, cell division, proliferation, metabolism. So we thought that, is there a possibility for us to then start to find whether there is a correlation? So if people are described as Vata, would we look, if we look at people who are of Vata kind, would they have differences with respect to this from the other two group? And could we then try to bring a molecular kind of a interpretation or subtitle to what are Ayurveda descriptions, and then can we start to think whether we can integrate it in modern settings? What is very interesting is Vata, Pitta, and Kapha also talks about how this vary in the lifetime of an individual. These are also very vividly described. So, for example, this shloka talks about how Vata, Pitta, and Kapha gets to be in a person because of the genetic component, Shukra Shonit Sanyoge. Then there's something like a prenatal environment, which is called Garbhashay, Matur Ahar Vihar, what the diet of mother is. And then it is governed by Jati Prasakta, ethnic background; Kul Prasakta, what runs in the family; Desh Anupatini, what is because of the geography; Kala Anupatini, what is because of the time; Vay Anupatini, what is because of the age; and then Pratatmanyata. So this is like written in 5,000 years back. So the science would have evolved even more earlier. But it is very interesting because in modern sciences, these are all different subjects which are dealt in a very fragmented manner. These are not all dealt together, and multi-omics is what aims to integrate all these sciences, right? So way back in 2001, we thought we will test it out. So we developed a working hypothesis that if these are the components which are described in Ayurveda, if we take people of these kinds who are at the extremes of each of the Prakriti type, would we get differences with respect to these kinds of processes at the level of expression or at the level of genes? We decided to test it out. And because we were developing all the other kinds of maps, like the Indian genome variation map, so what we said was we will take people from one genetic background and classify them on the basis of Prakriti and study them at their genetic and expression level and try to do a synthesis to see whether there are differences in the molecular makeup if you classify them on the basis of Prakriti. So this is what we did, cutting a lot of stories short. But if you look at any biochemical test, you normally have a range, right? But what we find is Vata, Pitta, and Kapha also has a range within the range. That means to say that supposing there is an X lipid profile, we take all of the people to be, if they are within this range, they are not disease. But what turns out that if you classify people by Prakriti, then they have a restricted range. So for these people, actually moving out of this zone is not a normal state for them. So that shows that all the normals are not similar. There are people who might be different levels might cause a disease to them. And as a result of which, we should not wait for all of them to come to another stage, because then this person might have evolved to the second stage of the disease or third stage of the disease. So that is what we've been finding over the last 20 years. We've been studying these Prakriti types in different populations, that Vat, Pitta, and Kapha differentiate with respect to many processes like blood clotting, cell cycle. It differentiates Vat, or Pitta is differentiated on the way their hemoglobin levels are, their immune responses are, and Kapha also can get differentiated on the basis of the lipid profile, zinc levels, etc. What's very interesting is that, as I described, that Vat regulates cell division is also a shloka. Parmanu bhed na parisankhya is a Vayu karm. And this is something which we observed. But this was like way back in 2008. So recently we thought that if this is we see in the peripheral blood, would we see them in their if we grow their cells? So if we take Vat people, Pit people, or Kapha people, and we take out their cell and we immortalize them and try to then look at their cellular properties, are they different? And we find, and this has just come very recently, that the doubling time of Vat is much higher than Kapha, and Vat and Pit are more similar, but Kapha gets differentiated with respect to their doubling time of cell proliferation. Now, the thing is that if they are different, the cell proliferation is also a condition of cancerous condition, but cell proliferation is also important in many physiological processes. So we asked a question, why do their cell proliferation become different? Is there a design principle behind them so that healthy individuals of Vat have more higher cell proliferation rates than Kapha? So we decided to test them out. So what we did was we give them a UV exposure, because that's a DNA damaging condition, and cells will have to recover from there. So what we find is that if we give UV damage, then Vat dies more. They have more dead cells than live cells compared to the others. But if we wait for some time, then after recovery time, all of them have really equal numbers. And that's because Vat has a growth advantage. So the time it takes for them to make doubling will actually bring them back to the same level. So it seems to be that inherently, if they are more predisposed to DNA damaging, then that might actually make them more susceptible if they keep on dying. So cell proliferation is a way they protect themselves. Recently, people are actually seeing that there are clinical conditions which are having a cell proliferation phenotype, just like between Vat and Kapha, what we find. And maybe there is a possibility that lithium response in bipolar, if they correlate with differences in cell viability, then we can use this Prakriti as a way to stratify people and see who are likely to have better response in different conditions. We've made other discoveries. For example, we find that if we classify people on the basis of Ayurveda, we find that they differ with respect to oxygen response in them. And then we find that such kind of a difference becomes relevant to which people can be actually more habituated to high altitude condition versus which are the people who can do better at sea level. Not only that, we could also show that if you have asthma and there is a condition of hypoxia, then there could be differences in people having an inflammatory consequence in asthmatic condition. So many such things we find from Prakriti knowledge. So the idea is that if you classify people on the basis of Prakriti, you get to see such kind of molecular axis, which then becomes very, very relevant in trying to predict who are the people who might do good in low oxygen or better oxygen conditions. Not only this, then we go back to literature. Ayurveda literature, and we find that Ayurveda also describes who are the people who are likely to do well at high, or who are the people who do well at low altitude conditions. So this is one of the kind of things which we can discover using the knowledge of Ayurveda. So this is what we kind of developed a framework that if you look at people by their feature, like look at Prakriti of people, then you will find that you can discover such things that high altitude adaptation, oxygen, management of oxygen differs between Pitta and Kapha phenotypes, and that becomes relevant in causing diseases of like asthma or thrombotic outcome in hypoxic condition. And then we can actually use this platform to see whether in Ayurveda, if they use a therapy for asthma for modulating Pitta Kapha, does it go to hypoxia? So this is what a student of ours tried. So he took an Ayurveda formulation which is used in asthma for Pitta Kapha modulation, and we find that it actually alleviates hypoxia. So this is what we call as Three Sutra framework, that the same dosha is the cause of a disease, could be explaining phenotypes which differentiate, and could also be a way to find recovery methods by Ayurveda principles. And hypoxia and hemostatic outcome, which we have seen, is now coming very relevant in COVID. Everybody has used a oximeter, and everybody is measuring oxygen levels, and not everybody responds similarly. And there are lots of inflammatory consequences if you do not control low oxygen conditions. So maybe in integration of such kind of methods might allow us to detect people who are differently responsive. And also maybe hypoxia management then can be governed by drugs which are used in Ayurveda, like Adadoda Vasika. We showed that how it could be useful in COVID-like condition, and this is now being tried out in a drug trial. So I'll get off from this, and I'll just say precision medicine does not seem to be new in India. It has an advanced approach to health management, and it can actually provide solutions for disease and wellness management. Just aside, because we had this yoga, and we all kind of seem to question sometimes that whether our systems are really working that way, or are they religious dictums and things like that. But you can see this is a whole group of people from US. It's a paper which has just come this month. They have done actually inner engineering, which is used by Sadhguru. They looked at their genomic study, and they find that there is a immune system modulation which you can do by following such kind of meditation, intensive meditation. And this came in a very prestigious journal, where they showed that how if you do this kind of meditation, then you reduce the cell cycle. You can see now how Vat could be possibly reduced. You can regulate immune response. You can reduce oxidative stress. You can have many more other kinds of things done, supposing you follow this. And this, they showed that how this meditation, then the kind of genetic networks which form in meditation are all disrupted during mild and severe condition based on this kind of color of red. And this is just basically to put in the perspective that how the science, method of science, could allow you to address such kind of questions, and then maybe we would have much more options for health management. And that's what we are moving towards in IIT, that how we can develop a transdisciplinary center for precision health using data sciences and combining many different systems of medicine and having a transdisciplinary approach. And that's where we recently got the sanction for development of an Ayur Tech initiative, a center of excellence at IIT Jodhpur, which might allow us not only entrepreneurship options, but provide us a new way to look at how to manage health and prevent diseases. So the idea is how we combine principles and practices with data sciences, engineering, and multi-omics method, and develop such kind of tech advancements to be able to kind of link Sanskrit with modern medicine descriptions of Ayurveda and modern medicine, integrate it with the new data, develop a lot of AI ML based models for large scale applications. So I'd like to thank you. This has been a 20 years journey. I just moved to IIT and lots of people from different domains have participated in this whole collaborative network, and the whole thing I developed with the Dr. Bhavna Prasha. She's an MD in Ayurveda. For 20 years, we developed this area, which is now called as Ayur Genomics in the country. And also there's been a lot of modern medicine people and all who've worked with us. So thanks. Thank you for giving me a time to present this work, and maybe if there are questions, I would be happy to address.
Thank you, ma'am, for very amazing talk. And, a lot of exciting things and a lot of exciting findings. Let me see if there are any questions from attendees. So you can either raise your hand or you can write in the chat box. Ashish, can you move Mr. Mahesh Kumar to the panelist so he can ask?
Hello, good morning, ma'am. Hello, am I audible?
Yes, yes, you're audible.
Yeah, thank you for your nice presentation. It was really informative for me because I am from the engineering background. I have no knowledge about genes at all. So it was completely new for me and I enjoyed the session completely.
Oh, thank you so much.
Right. So, ma'am, I was surprising that how the genes carry disease information from parent to offsprings. So which part of the genes do they really carry, or we have just notion of genes they carry diseases from one generation to the next?
So we have all the genes like I showed. These are all genetic material which are passed on every generation. So we get one copy of the entire set from mother and the other copy from the father. So if there is a defect, supposing in the paternal lineage or in the maternal lineage, and if that chromosome copy has come to the next generation, they would inherit it. So there are laws governing such kind of inheritance. So it is not that every time they mate, the same one would come, because there are also genetic recombinations in the parents' gametes at the gametic stages. So, but if they segregate, if they pass on that defective copy, then surely the next generation might get it. But again, it would depend on whether one copy is sufficient to cause the disease or you need two copies to be defective to cause the disease. And that's why it's very important. If once you sequence the genome, you need to first know the variations, whether those variations are likely to cause diseases, and the variations in single copy or two copies will cause the disease. And then you sequence a person who is diseased and try to follow their maternal and paternal lineage to see if it runs in the family, whether every affected person shares the same genetic makeup. Then you will say that that person could likely have the disease because of this change. Such kind of information becomes very, very important in families who have rare diseases, because the burden of rare disease is immense in the family. It is very crippling. And if you have such kind of an information in the families, then you could prevent by doing some prenatal testing, of course, following all ethical procedures. And that's why these things are coming to be very important. So it's not that every disease has a genetic origin. Rare diseases mostly might have a genetic cause because they are early onset. But if you have a common disease like diabetes, asthma, it tends to run in the families, but there is also an environmental component. So it's like a car with a weak tire. If you take it to a bad road versus you take it to a good road, then you might have a better management of the car. And that way, the body has genes which are primary, then there are genes which modify, and then there is an effect of environment which work through the genes. So all of it combination will tell you the health state and the disease state. I don't know whether it answers the question. So
Thank you, ma'am. Actually, I wanted to know that is there any data set which contains gene sequencing and the disease name which they are carrying, so that
yes,
we can, yeah.
yes. So there is a site called OMIM, which is called as Online Mendelian Inheritance in Man. So many of the genes which are associated with diseases, you will find information by either giving the gene name or giving the disease name, but that's not the only resource. There are huge number of resources now available where one can find such kind of information. So I'm sure during the time of this workshop, maybe there would be some kind of an orientation towards that.
Thank you, ma'am. Thanks a lot.
Thank you.
Let me check if there are any other questions. Okay, if there are no any further questions, then we can close for today's session. So the first half session. So we will have session 3 at 3 p.m. starting one titled Cancer Treatment Using Multi-Omics by Professor GPS Raghava from IIIT Delhi. So we will meet again at 3 p.m. And thanks for joining these two sessions. See you.
Thank you.
Hello, are you there?
Yes, good afternoon, sir. So Rajveer, over to you. Maybe you can start. Sir, we will start in 1 or 2 minutes.
No problem.
Sir, am I audible?
Yes, yes.
Oh, okay. So, good afternoon, one and all. So, it's really an honor for me to introduce to all of you to our next speaker, Dr. Gajendra Pal Singh Raghava, who is going to give a talk on cancer treatment using multi-omics. Dr. Raghava was born in Bulandshahr, UP. Dr. Raghava is currently a professor and head at Department of Computational Biology, Indraprastha Institute of Information Technology, Delhi. But before joining this institute in 2017, he was working as a chief scientist at Bioinformatics Center, CSIR IMTech, Chandigarh. Dr. Raghava did his MTech from IIT New Delhi in 1986 and then PhD in Bioinformatics in 1996 from IMTech, Chandigarh. He worked as a postdoctoral fellow at Oxford University, Cambridge, UK. And then he also worked as a bioinformatics specialist at UAMS USA in the year 2002 and 2003, where he established the bioinformatics infrastructure. He also worked as a visiting professor at Postech, South Korea in 2004. He was awarded a National Bioscience Award in 2006 by Department of Biotechnology. His contribution in the field of drug and vaccine uh, candidate has been recognized by CSIR, and he was awarded Santi Swarup Bhatnagar Award in the year 2008. His contribution in the field of computational resource of drug discovery was recognized by Department of Science and Technology, and he was conferred the prestigious J.C. Bose Fellowship in 2010. Currently, his group is mainly working on genomics-based solution for understanding and managing treatment of disease, particularly cancer. And he was awarded Scientist of the Year in 2019 by Organization of Pharmaceutical Producers of India. His group has developed more than 250 web servers and 40 databases in the field of computer-aided drug and vaccine design. And with over 19,000 citations and an H index of 73 and a G index of 195 for his publication. So please join me in proudly welcoming Professor GPS Raghava. Over to you, sir.
Thank you very much for nice introduction. Now I will share my presentation. Okay. Is my presentation visible? Can anybody confirm?
Yes, sir.
Yes, sir. You're audible and your presentation is visible.
All right. So today I will talk about the computational resources for managing treatment of cancer in the era of multi-omics. So as you can see, the major focus will be on multi-omics, where I will talk about the different type of omics, how we can use them for managing or treating the cancer patients. I will mainly focus on the tools or resources developed by our group. It's not practically possible to cover the all groups, so but we have written the review. I will just give the reference of the review where we have compiled all the resources in the field of this computer aided cancer treatment. So if you are new to this area, then I will prefer you to go for this website. This is my website. All the resources, tools, or mobile app developed by our group are available on this website. So this video will be available to you, so you can search anytime. Even if you don't want to note down this link, you can go to the Google and put Raghava GPS, and you will get all the links, all the tools related to our website. Though I know it that you have already read about the multi-omics, you know the different type of omics, but just for more clarity and for revision and giving you some idea about the different type of omics again. So we all know that this is basically any, let's say for example, human have the organs or tissues, liver, lungs, or muscle. These are the different tissue. Each tissue is made of cells, and cell is considered as the basic unit of living organism. We cannot have the living organism less than a cell. There are a number of living organisms which are single cell based living organisms, but you cannot have less than a cell. So that's why cell is called the living unit of we call the small unit of living organisms. Like for any matter or any material, the basic unit is atom. Same way for living organism, basic unit is cell. Cell have the, I'm talking about the human or eukaryotic cells. In eukaryotic cells, you have the nucleus. In this nucleus, there is a genetic material. That's called the genetic material. That's why I called human genome. So human genome is nothing. It's a basically genetic material, which is basically transferred from parents to child. That's a common thing. It means all the genetic related information is there in the nucleus. But it's not randomly available. It's well organized. In case of the human, it is organized in chromosome pairs. In case of the human, there are 23 pair of chromosomes. One pair is dedicated for the sex, and rest of the two is for the other jobs. And this chromosome is nothing. It's a basically organization of the DNA, because genetic material is there in the form of the DNA. A, T, G, C. You know that all the DNA is made of 4 nucleotides, A, T, G, C. And it's a long tube. So in human genome, there is around 3 into 10 to the power 9, 3 into 10 to the power 9 base nucleotides. Why we call the base? Because DNA is in the double helix form. So it will be base pair. Instead of 1, there will be 2 nucleotides. Complementary strand will be there. But if you see the total number of nucleotides in a single strand, then it will be 3 into 10 to the power 9. All right. Then this DNA material is wrapped around the stone. That's basically proteins. And the modification of these histones is called the epigenomics. This study where we try to understand the methylation or post-translation modification of DNA binding proteins, that we call epigenomics, right? And the whole DNA, if you take the whole DNA, this is called the genomics. Whole genome, or the study is called the genomics. And as I told you, this is found in form of the double helix and made of 4 nucleotides. But whole genome, 3 into 10 to the power 9, is not involved in making the genes. Is not involved in making the genes. Only few regions which are responsible for coding for the proteins is called gene. So these are called the transcript also. So if corresponding to gene, it translates into the mRNA, and number of mRNA copy of these genes is called transcriptomics. So transcriptomics is nothing, it's a basically number of mRNA corresponding to all the genes is called the transcriptomics, their copies. And this is also called, in simple word, gene expression. We say if we study the gene expression of all the genes expression of all the genes in a cell, then this is called transcriptomics, or this is called the gene expression studies. So these genes or this mRNA translate into the proteins. And if we consider all the proteins in the cell, then this is called proteomics. Proteomics means all the proteins in a cell. And again, like DNA is made of 4 nucleotides, same way the proteins are made of 4 natural amino acids like alanine, valine, cysteine, aspartic acid, like that. There are 20 natural amino acids. So this is protein is nothing, it's a basically polymer of 20 natural amino acids. Then there is a post-translational modification where the proteins have been glycosylated by different sugars, or there is a phosphorylation is there. Then this study is called glycomics or phosphorylation. And similarly, lipids can also be connected with the proteomics. Then lipidomics is called. If we study all the lipids, it's called lipidomics. If we take the metabolites generated in a cell, then this is called metabolics. So these are the major words commonly used day to day in the area of omics, or this is called the multi-omics. Now, question is, a lot of talks are there on the personalized medicine. So in personalized medicine, normally we study the family history, means whether there is a cancer in the family or not, whether the person family has a history of some other disease. So that's the one part. Proteomics, all the proteins, imaging systems, metabolics. So all parameters we can use for the personalized medicine, basically integrate all the information. This is what personalized medicine. In simple terms, if we go to the pharmacogenomics, that's a very commonly used term nowadays and heavily used, where the study of variants of the DNA and variant characteristics as related to drug response. Why we call pharmacogenomics? Because pharma is a one area where we study the pharmaceutical studies, and we try to relate with the DNA change and RNA change. So what are the major areas where we can relate it? DNA sequence. If there is any mutation in the DNA, whether it will affect on the drug response or not, or any mutation is responsible for a particular disease, sort of things. See, another part is gene expression, where we study that which gene will be expressed, which gene will not be expressed, or which gene will be overexpressed, or some gene will be underexpressed. So that's area also connected with the drug discovery. So we can consider that. Another is copy number. Sometimes number of genes, copy in genome, it's not single copy, it's a multiple copy actually. So it's very important how many copies are there. Because if you have number copy, the advantage the genome has, that if one copy is defective, the other can take care of it. Same way, there is a mutations in the genome that is called SNP, single nucleotide polymorphism. It may be synonymous or it may be non-synonymous. Synonymous means amino acid will not change. In non-synonymous, amino acid will also change. How we relate with it, depending, one is called pharmacokinetics. In case of the pharmacokinetics, we try to understand the individual difference in drug metabolism, because all drugs are not suitable to all the persons. Some drugs are suitable to most of the persons, but some drugs are toxic to some particular type of population. So that is basically due to the drug metabolism. If drug will not be metabolized, it will be there for the long term in your body, it will be toxic. So we study in pharmacokinetics. And the response we try to understand is the pharmacodynamics, okay, where we understand the physiological drug response, what will be the efficacy, toxicity of the drug, identify whether it will be respond or not respond. So these are the major studies. So that's why a lot of people in the pharma nowadays, they are jumping to the pharmacogenomics rather than staying in the genomics. Same way the persons who are working in the drug discovery, they are jumping towards this pharmacogenomics, or they are relating the drug discovery with the genomics. So this is one of the major questions I face from the most of the traditional scientists, or maybe let's say old scientists, old school of thoughts. They still feel these are the basically big words like personalized medicines or all these things. Actually, they don't believe too much. Actually, they believe this is their only matter of research only. There is no practical use. So what I did, I take the latest slides from the FDA. If you don't know, the FDA is a Food and Drug Administration from the USA, where most of the drugs, if you want to sell in the US market or number of international market, the drugs will be first approved from the FDA, and then it will go to the market. So I have taken that slide, and this slide shows that in case of the oncology means cancer, 140 drugs, they are advising not to give to the patient till their biomarkers is checked. Biomarkers is in the sense that it is suitable to that person. What happened in the case of the cancer? Sometimes the cancer patient is already weak, and if you give the drug, the cancer patient will die due to the reaction of the drug. Why? Because it is not suitable to the person. So it's important to give the drug after checking the certain type of biomarkers. And this is the basically name of the biomarkers with the color that which biomarker works for color. Okay. So this is the just and it's so that for oncology, this area of drug biomarkers is important. It means you have to check for the individual whether the particular type of protein is there or not. If it is there, then accordingly we have to give the drug. So these are called drug biomarkers. They are very important. So here is some examples actually. Let's say take the example of Chigan. That's a basically drug normally given during the treatment of the HIV patients. But it has been observed that if the patient carries a particular allele of HLA allele, then this may experience the hypersensitivity. So it is advisable before prescribing this drug, one should check HLA-B 5701 in advance. And if the person carrying this HLA, then this drug should not be given to the person. Okay. There is another type of oral metabolizer, and this drug is given to the patients suffering from the heart disease, cardiac disease. So it stops to interaction between the platelets. But it has been seen that this drug is metabolized by this particular CYP450, because you know in your liver, most of the drugs are metabolized by the cytochrome 450 alleles. And one of the dominant allele is CYP2C19. Okay. If it is not available or it's not expressed in the particular person, then what will happen? Then this drug will be highly toxic. In that case, it is not advisable to give this drug. We have to think about another drug for this purpose. P2Y1, which will inhibit the platelet rather than stopping the interaction of the platelets. Basically. So idea here is, with the time, we will have the genomics, biomarkers. It's already there, but with the time, many biomarkers will come up. So that you have to check the genomic profile of the person before prescribing any drug to the person. So that's very important. And particularly in the case of the cancer, it's more important because person is suffering from the cancer, and most of the cancer drugs, they are toxic in nature. Because the problem here actually that normal human cell is similar to the cancer cell. Because cancer cells are originated from the human cell. So when you give the drug to kill the cancer cell, the chance is there it will also kill the normal cells. So it's better to have the different type of genomics based biomarkers for prescribing any drug to the patients. This is a server we developed for these number of isoform of CYP450. Because these 5, 6 isoform, they metabolize more than 90% of the drugs. So we have developed a model where you give your drug or maybe some chemical which you want to take as a drug candidate to our server. Our server will predict which isoform will metabolize it. If that isoform is not available in the person, then that drug should not be given to the person. That's the advice. Now we have to go different type of omics and advancement in the field of omics. So there are the two major techniques used from last number of years. One is called cDNA chip or cDNA microarray, which measure the comparative expression between the normal and disease case. Okay. And there is another technique. One is, let's say, chip technology. That's a Affymetrix, where it measures the actual expression of the genes. The real that's basically called the absolute expression. Here it measures the comparative expression. These two technologies are quite old. This was developed in the 95. This was developed before 95. So they are old technique, but heavily used so far. But 2008, there is a major change in this area. The technology come called RNA-seq, or this technique is called next generation sequencing. Initially, it was evolved to sequence the big genomes. But later on, this technique becomes so popular, people are using this technique for measuring the gene expression or transcriptomics. As I told you, transcriptomics is nothing. It's basically called expression of all genes in a cell at a time, or maybe number of mRNA copies in a cell at a given time. That's the definition. So this technique is used nowadays for the gene expression, because it not only measures the gene expression, it also measures the mutations or insertions, deletions, all sort of things. It's a do the basically gene sequencing technique. But it becomes so cheap that people are using it for the measuring the gene expression. So this is the concept of the cDNA technology, where you take the normal samples, normal cells, and tumor samples, and extract the mRNA. mRNA is the number of copies that's basically corresponding to the gene expression. Convert it into the cDNA, complementary DNA, and then attach the particular dye, which is, let's say, green color. And for the tumor samples, again extract the mRNA, convert to the cDNA, and attach to the dye, which is called CY5. Okay, C5 dye, and this color is red. Here in this chip, in different walls, there are complementary DNA corresponding to the cDNA, corresponding to the genes, because we know the reference genome. And then you will mix the two populations, this one, and put it here. Depending on the color in the plate, we normally say whether the normal cells, the particular gene is expressing more, or in case of the tumor, it's expressing more. So you can measure the relative expression level based on the color. If red color is, let's say, more dominating, or its amount is more in a way than the green color, then we will say that the particular gene is overexpressed in case of the tumor. Then Affymetrics have a coding of the each gene's representation here, and again we take the mRNA, convert to cDNA, label it, and after fragmentation we will put on the chip. So that's a will measure the actual expression, because their biomarkers are already labeled on the chip. So these are the basically general techniques. And nowadays, next generation sequencing is very popular, where we take the mRNA, okay, then we convert it into the cDNA, then we make the expression sequence tag library, and then we have the short seq reads, basically sample preparation. Then we make the number of copies and break into the pieces. What does we do the pieces? That this sequences. So then there will be a lot of fragments. There is all the small fragments, and then will be used for the sequencing. And this is basically, once we got from this one, we got the small. We will read it, and this will call the small reads, basically. And then we will make this reads on the human genome using the different softwares, okay, and then we count how many reads are mapped on a particular gene, how many transcripts are mapped there, and based on that we will count the read table. Then it may be fragments per kilobase of transcripts per million mapped reads, or reads per kilobase millions. These are the different terms. These are the units for measuring the gene expression when you get the data from the RNA technology. Right. Nowadays, there is another advancement is going on. A lot of people are working, because whatever the work we are doing earlier, that's basically average of all the cells. It may have the different type of cells, because you are not separating the different type of cells. It is possible that the population which you are sequencing may have some immune cells, maybe some other cells, okay. But we want to know each type of cell separately. So nowadays techniques are there where they take the tissue sample. From tissue sample, they are segregating the different type of cells. Means they are separating them each other. So then one type of population will be in the one type of tube, another type of population will be another type of tube. So you have the each type of population. Okay. Then you do the different type of sequencing, expressions, and all sort of things. So you will get the how many, what is the percentage of different type of cells in your samples, and what is the basically type of expressions or sequencing is there. So that's because earlier traditional techniques, we take the bulk level of RNA, we take the average gene expression, and then we compute the heterogeneous other parts, which was not practically possible. So single cell technique is becoming popular day by day. So let's say if you're experimental biologist, that's fine. But if you're not experimental biologist, if you are in the biotics, then you need the data, right? Data means Some database should be there from where you will access data. So this is one of the repository. One of the repository where you get all the gene expression related data. So where scientific community is submitting and it is freely available, you can curate it, you can browse it, query, analysis, all informations are there. So this is one of the major database. If you are really interested in the gene expression based work, then this is one of the data actually. Okay, I'm not going into detail. There is another database. This is called ArrayExpress database, maintained by EB. This also provides the samples related information and gene expression. If you are interested in single cell, then you can get the data from this repository. Why I'm telling all these things? Because if you want to work in the field of biometrics, it's important for you to understand wage data. Because no data, no bioinformatics. No data, no biometrics. So it means most important part in biometrics is data. So if you get the data, then you can do the bioinformatics. So this is a repository database which provide all the information related to single cell gene expression. So if you are interested in single cell genomics or single cell work, then you should go for this website. Now, I will come on the cancer patients, because so far, whatever I talked to you is a general process of genomics. This is important to understand before we go for the cancer.
Sir, sorry to interrupt you, but could you please make it full screen? Someone has commented that it is not full screen.
It's full screen from my side actually. It's not coming on your screen.
So it is not in a full screen because it is not clearly visible to students. They are commenting that the screen is not full screen.
Okay, I will share it again.
You are muted. You have to unmute. Somehow you got muted.
Now you are getting on the full screen. Raj,
No, it's still the same.
it's not showing on the full screen.
No, it's just showing the small window, not the full screen mode.
Okay, just hold on. I'm not getting why. Okay, I will do one thing. I will go on the whole screen. Just. Are you getting now on the full screen? Can
Yes, sir.
you confirm?
It is working, but we are not able to see the slides. It's web browser is being shared. Maybe you can try to share the slide window.
I will share it again. Are you getting now?
Sir, again, it is the browser is being shown.
You are not able to see my screen?
We are able to see that the different windows, but not the presentation. Maybe you can move to the presentation.
No, I am on the presentation itself here. Not getting why there is some problem. Some participants write that they are getting full screen now.
Yeah, we can see the full screen, but your slides are not visible. We can see that welcome to NTCP 2.0, that some other window. The tool NTCP we can see, but not the slides.
Just hold.
Sir, you can try on the presenter view, that tab, if you can see presenter view on the left side.
On my side, I'm not. Try now.
Unfortunately not, sir. Yes, sir, now it is working.
It's full screen now.
Yes, you can move the slide to the one that you left last.
Okay.
Thank you, sir.
Okay, so I was talking about the cancer patients. So first thing, when any cancer patient is there, you have to do the screening or diagnostics. If you go to the traditional techniques, there is a CT scan, MRI, PET, all type of techniques are there. After that, you confirm that person is suffering from the cancer. The next is risk assessment or prognosis. Means in what stage the cancer is there, or how long the person will survive. Okay, so that is called basically cancer staging, cancer, different type of cancer biomarkers to understand what is the life of expectancy is there, because normally the patient doctor says that maximum that cancer patient will survive for 1 year, 2 years, 3 years, whatever it is, and based on that prognosis, we give the treatment. Now, question is again treatment. All drugs are not suitable. Then we have the different type of biomarkers. So these are biomarkers are basically mostly traditional biomarkers. But in this talk, my major focus will be on the genomics-based biomarkers or omics-based biomarkers. So when you talk about the omics-based biomarkers, or we talk about the omics, then we have to understand the availability of the data or technology of the data, technologies available for that particular. Let's say this is a patient. This is a patient. So one data which is available or you can generate is genomics data. Genomics means DNA of the cell. ATG, CTG, like that actually. And if you take the whole genome, for example, 3 to 10 power 9, then this is called WGS, whole genome sequencing. But sometimes you are not interested in the whole genome. You are only interested in the genes which code for the proteins. and that is called the transcriptomics. Okay. So this data is called the whole exome sequencing, because your gene is made of intron and exomes. So we are taking whole exome sequencing, means whatever the exomes are there in the genes, we will sequence them. So this is technology. Why we go to the WES? Because it is much cheaper than the WGS, as well as size will be much smaller than the WGS. So this is the one based on the sequencing. Another is transcriptomics. Transcriptomics means where we want to understand the expression of the genes. Means some genes will be overexpressed, some genes will be underexpressed in certain disease conditions. So based on the gene expressions, we can measure the different factors. Similarly, there is another study, and this technology here, the transcription sequencing is RNA sequencing. Or there is another technology called epigenomics. These are the sequencing technologies. In epigenomics, we try to understand the binding of DNA with the histones, basically, or histone modification, methylations. Lot of things are there related to epigenomics. So these are the major point where you have to perform the different type of data, like a whole genome. You have to understand the mutations, small insertions, deletions, copy number variation, structure variation. These are the major points you have to study when there is a whole genome. Same way in case of the transcriptomics, you understand the differential gene expression, what happens when you give the particular drug, whether the certain genes will be overexpressed or underexpressed, sort of things, or whether there is an gene fusion is there, or also a possibility that alternate splicing is there, because there is intron and exons, and sometimes there is a jumping of the introns basically. So that is called alternate splicing. So RNA editing. So all these factors basically. In case of the epigenomics, we measure the methylation, histone, blah blah, and then we do the study. So this is the overall idea about the different type of omics or multi-omics basically. If you are really interested in the cancer genomics, excellent resource for this is GDC, Genome Data Commons, and under GDC there is a one section which is called TCGA, the Cancer Genome Atlas. This provides excellent data. In fact, most of the biotics in last number of years, they are using data from TCGA. So TCGA is become a gold standard for this type, right? So I'm not going to the detail of the TCGA. You can read more about it, but it is one of the old data set, though they are ending this project, but data is available. You can work on it. Now, I will talk about the resources. Just hold on. Ganesh, I am on the meeting actually. Yeah. Okay. Okay. So, computational resources for cancer research. These are the resources created by our group. These are not all resources. We have created many resources in cancer research, but I am telling you, or I am just describing you few major resources which our group generated in last few years. Because we have a different way of thinking. Most of the biointelligence, or particularly in developing countries, we use the data created by the developed countries. But our group is involved in developing our own resources so world can utilize it. So here I will talk about only few major resources. So these are the two databases, Cancer DR and Herceptin created for the understanding the drug resistance in cancers. Similarly, we have the other resources, which are the gene databases, like pancreatic cancer or other cancers related databases. Then there is some databases which can be used for the drug delivery. So we call the virus TDB and tumor hope. These are some databases which is very useful for the treating the cancer patients. Then there is other cancer biomarkers. So I will cover about these databases. These are the important resources anybody working in this area. So when we enter in this area first time, I think we started this area in 2012. First work we have done is cancer creating the database that we call cancer drug resistance database. Why we created? There was a lot of talk, number of publications was there, where they demonstrated that in cancer have the drug resistance, where it worked for some time and then it becomes the resistance. So in order to overcome this problem, there was a major studies where they demonstrate the effect of the drugs, different drugs, on different type of cancer cell lines. These cancer cell lines are different, corresponding to different type of cancer cells. So we created this database, and this database provides the information on 952 cell lines, and these are the cancer cell lines. On these cancer cell lines, 148 drugs have been tried and checked what will be the IC50 value. As well as these all cell lines have been sequenced, their mutations have been identified, copy number variation have been checked, gene expression have been checked. So this was complete information. The goal was basically how to find out the relation between the gene expression of a cell, of a cancer cell, and its resistance for a particular drug. Okay. So this database has been created a long time back, and it has the information from CCLE, COSMIC, all the related information. Not only the information, we have already checked the drug targets of these cancer drugs, and then for each target, we predict the structures, we create the alignments. The overall aim is to help the community, scientific community, to work on the cancer drugs. Then there is a Herceptin R, Herceptin resistance database. This is a database which is very specific to the particular drug. I don't know how many people know about it. In case of the breast cancer, one of the commonly used monoclonal antibody for treating the cancer patient is Herceptin. And Herceptin, because it's a monoclonal antibody, it is a costly business. So I don't know the cost now, actually. I know a few years back, the cost was of each dose was nearly 3 to 4 lakhs. So even if the cancer patient takes the 4 doses, it means there will be 15 to 60 lakh, around 15 lakh rupees will be there. And if it is not working, it will waste the time of the cancer patient as well as economic loss to the patients. So it's important to have the biomarkers where it checks in advance whether this antibody will work or not work. Because in certain patients it works, in other population it doesn't. To understand this part, we collect and compile the information from the literature and other resources so that we can set the relation between the resistance or Herceptin and the gene expression of the cells. Similarly, we go for the database. This is called pancreatic cancer methylation database, where we have measured the gene expression, methylation related information of the genes basically. So I'm not going into the detail. All are available. You can use them. Similarly, there is another database called a database of epigenetic biomarkers, epigenetic biomarkers, where you can find out the epigenetics. Epigenetics in case of the cancer patients. We have taken all the genes which are involved in the epigenetics, and complete information has been provided in this database. Please remember one thing. These databases are all peer reviewed. They are all published in lab journals. So you can use them without any doubt. It's not like that we are developing the database without maintaining an international standard and all sort of thing. These databases, they are maintaining international standard, published in the top level journals, so that we also get the feedback that standard or quality of the database is there. One good part we have actually. Most of the resources developed by the bioinformatics community have been down in next 5 years. But all our databases, web services are working continuously since we start our group. No service is down so far. This is very interesting work. The idea came from my students that the virus can be used as a treatment for the cancer. So these are called the oncolytic viruses. If you remember, even in the Covaxin, in the Covishin, the drug is delivered through the adenovirus. The virus can be used to deliver the drug. Okay. So here, these are the virus which can be used to treat the cancer. And we have collected all this information. So you get the complete information from a single source, and this database, OVirus TDB, is available free to use. So you can use this database. So this is basically how many records are there, how many research articles there. So I'm not going in the detail. Then we created another database called Cancer Liver. In this case, what we did, already a lot of work had been done in the past on the cancer liver. They identify the biomarkers based on the proteins, based on the mRNA, based on other molecules. What we did, we collect and compile this information and we put it in the form of the database. The question you will ask, where is the science? You are just collecting and compiling and putting the information here. I will tell you. In order to create a database, my students take years to collect and compile the complete information. And you are getting that information. You are getting the information on the single click search. So that's called the facet to the community. So we are saving a lot of time of the community. They can also search it, they can also find out, but they will waste a lot of time actually. So instead of, let's say, 1,000 people work for that searching, only one person is doing the job. Another advantage is, once my students or me work on any database, we get the depth knowledge of the field. After that, working in that field is becoming an easy job because we know what has been done so far or not. These are the major concerns why we are creating the resources. There is another database which means in the enhancers, because you in the transcription side, there is a transcription motifs or enhancers where the transcription factor binds, and based on that gene expression started. We have compiled all the information from the different databases like Phantom, Cosmic, and other things, and we map it with the cancer patient. Okay. So this database, you can use it for your work. And this is a website. So one part is database where we work. Another part is prediction. Fine. We collect the information. It is available to the public. Question is basically from user side. Can you do any predictions Can you identify new drug candidate? Can you predict the vaccine candidate? Sort of things. So in addition to the resources or databases, we also develop the web services for predicting biomarkers, different type of biomarkers. So here is a personalized drugs and vaccine site. I'm not covering everything. I'm covering the major sources developed by our group for drugs, vaccines, and biomarkers. So here there is a cancer talk, personalized vaccine drugs. These are the two database. No, not database, protection method. Then this is a drug discovery biomarker DIPSEL, cancer biomarkers, then peptide-based solutions, sort of things. I will cover few of them. This is very important to everybody want to have the vaccine against the cancer. There is only limited success so far, though there is immunotherapy which is used, but designing the vaccine against the cancer is a major challenge. How to work for the vaccine, that's a challenge. I will tell you that, look, for example, coronavirus. Within 1 year, we got number of vaccines against the coronavirus. Why we have no vaccine against the cancer in last number of years? The reason is very simple. These pathogens like coronavirus or any bacteria, fungus, they are foreign organism. Okay, our system recognizes it, and then we can easily design the drugs. But cancer cells are nothing. It's basically disorder of our own cells, or normal cells become the cancer cells. So how to discriminate the two is a major challenge. That's why we are not able to design the vaccines against the cancer. So here we propose genomics-based solution to handle this. So what we did here, first we analyzed the two databases, Cancer Provar and CCAD, and based on that we identify the cancer sensitive genes or frequently mutated genes. These are the two types of, because some genes there is mutations, but it is not leading towards the cancer. They are not cancer sensitive. But there are other genes, set of genes, where they are very sensitive to the mutation. Even single mutation may cause the cancer. So we identify 16 genes which are cancer sensitive. Where you have a mutation, then they will lead to the this called cancer. So we have taken them as a vaccine candidate. Now we know they are the vaccine candidates. Next person was how to identify or how to induce an antibody that only bind with the mutated region or mutated protein, not with the normal protein of the cancer vaccine candidate. So what we did, we generate the all possible 9 months, because we know for any immune system, for recognition, it is important that it has to bind with the HLA. And in case of HLA or MSC, class 1 or class 2, but here we don't need the full length, even 9 amino acids sufficient. So we have taken the all possible 9 amino acids have been generated. Then we check whether it is present in the reference protein or not. Reference means proteins in normal cell. If it is present, then we remove these peptides because we don't need peptide. So now we take the peptides, new peptides, which have been generated in case of the cancer and which are not there in the reference genome or in the normal cells. And we call them neopeptides because these are new peptides which are basically cancer recessive. They are only in the cancer patient, not in the normal human. Once we know that these are the regions which are mutated in the cancer but not in the normal, then we predict whether they can activate the different type of cells like B cells, T cells, or they will bind with HLA class 1, class 2, CTA epitope, sort of things. So we check it. So any peptide which can also activate our immune system or which can immunize our immune system can be a vaccine candidate, will be a vaccine candidate because it is found in the cancer patient but not found in the normal genome. So this is a major theme of this work. But sometimes you want fully personalized because reference genome is not representing any individual. It's a basically genome of average genome of all the persons sequenced so far. That is not giving the actual picture. So another way is fully personalized model. What is this fully personalized module? Let's, for example, take a cancer tissue from a cancer patient and healthy tissue adjacent to the cancer tissue, means they are this is a healthy region which is not infected, which is not cancerous. Then do the sequencing of this gene, this part of these cells as well as healthy cells, and generate the all 9 mers and do the healthy proteome mapping. If yes, if it is found in the healthy the peptide, remove it. If it is not, then this is a patient-specific cancer peptide. So this peptide is there in the cancer patient, and check now whether it can activate the different type of cells. So predict the epitopes, B cell epitope, T cell epitopes, and then you get a epitope promiscuous peptides that you can use, that peptide you can use for the immunotherapy as well as a vaccine. Immunotherapy is very popular, where you take the peptide, you immunize the cells outside the in vitro, and then give to the human being. Okay. So that is called T cell based therapy. You can use it. What our server will do, because the server is automatic in nature, once you give the genome, it will do all the work for you and you can use it. Okay. Similarly, we have another recently database that's very interesting work. What happened normally, we believe the HPV and some cancers are due to the virus. But this is not true. There are a number of cancers where we don't know how they generate their virus is involved. And it has been seen, for example, lung cancer, that in lung cancer sampling, if you do the sequencing, number of virus sequences found actually. It means the virus is involved in certain % of lung cancer patients. So can you do vaccines against these viruses? Because developing vaccine against the virus is easy. So we did the proposal that if we have the vaccine candidate or we develop the vaccine against these viruses, then lung cancer can be avoided in the future. So these are the basically servers for the futuristic. Maybe people will not understand for a few years because due to the lot of cost and other things, but in future it will be highly useful for the community. This is another interesting server for drugs. The problem here actually, if you go to the market, there are a number of anti-cancer drugs are there. It's very difficult for a doctor to decide which drug should be given first and then next. Means how you rank the drugs based on its efficacy for a particular patient. So here we propose the ranking of anti-cancer drugs based on the genomic profile of cancer patient. And this genomic profile may have the gene expression, copy number variation, mutation, like this. So here we have shown that these are the name of the drugs. These are the name of the cancer, name of the tissues. So that's let's say liver means liver cancer. Liver means liver cancer here. So how much this drug will be effective? There you can see the certain drugs like this one work for most of the cancers. Others are basically specific to the particular cancer only. Doesn't work for the other disease. So this itself shows that all drugs are not suitable to all the type of cancers. But that's a different type of cancer. So now you can do the mutations, variations, expressions. So we have done the prediction methods and we demonstrate that if we develop the hybrid model, that predicted ranking or actual ranking, there is a 0.78 correlation. So means it is practically possible. It's not 100% perfect, but certain extent we can rank the anti-cancer drugs based on the genomic profile of cancer patients. So it will be easy to manage and maintain the cancer patient rather than giving the random drugs to the patients. Give drugs based on the genome. This is another server for designing inhibitors. We know it, that how to design inhibitors. But the problem with the cancer or any type of cancer, even in the, let's say, pancreatic cancer, there are different type of populations which need the different type of inhibitors. Can we identify inhibitors which can inhibit the all type of pancreatic cancers? So that we call the promiscuous drugs actually, which can we identify with our models. So we have developed the number of models, and when you give any chemical, it will predict from different model, and then you say how much it will lyse the different type of cell lines of pancreatic cancers. So we have to select a drug which can handle the most of the cancers in pancreatic. This is basically server for predicting the HERCEP, this HCG, that's based on the large-scale transcriptomics data, gene expression data. We demonstrate that from the different platform we can use for the survey. Similarly, we have the biomarkers for predicting the stage of the cancer, because you can have the different stage, early stage, okay, advanced stage. There are different 4 stages are there actually. So here we have taken the discriminate the early and late stage of the cancer. This is important to understand the life expectancy of the patient as well as the life expectancy of the patient as well as to treat the cancer patients. So I'm not going into the each and every server detail, but these are different type of prediction methods which are designed for different type of stage of the cancer, or maybe sometime that let's say skin progression, skin cancer progression, how it will progress, local metastasis or distant metastasis. So all type of status you can predict using this server basically. This is based on the HLA, because all humans are not same. They are a different type of HLA actually. And based on the HLA, we can predict how risky it is there to the predict. This is based on the this cancer, okay, colorectal cancer. And in this colorectal, this is based on the 5 protein based biomarker. So far, I talked to you about the different type of biomarkers. The question is basically, can we think about a little bit about the therapeutic cells? So here I am just covering a little bit about the protein or peptide based therapeutics. So this database we developed to the users to predict the anti-cancer peptides. So this database has been collected and compiled from the literature, and we create a database which is basically identifying anti-cancer peptides. This method predicts the anti-cancer peptides. So let's say if you are interested in the field of the anti-cancer, then this will help you to design the better anti-cancer peptides. This is anti-CP2 which is basically version 2. These are the tumor-homing peptides. These are basically peptides which essentially go and bind to the different type of tumor, basically. So if you attach the drug with these peptides, they will deliver the drug on the drug target site. That's very important for managing any cancer. So this is for predicting the tumor-homing peptides. Okay, so far I talked to you about the cancer resources developed from my group. But here we write the recently review in this year that is called computational resources for identification of cancer biomarkers from omics data. Okay, you can read from the title itself. It covers all the biomarkers or resources related to the cancer have been reviewed here. This is a review, and this is fortunately that's basically editor's choice, and it is freely available. Otherwise, they put the cost on the publication. But this article is freely available to the community, so you can take the advantage. And it has a number of excellent tables which describe the different type of omics-based biomarkers. So thank you very much. That's all for me now. Any question from the audience?
Thank you so much, sir, for very engaging session, and it was really basic. I think everybody could have understood these things without even having background. So I would like to now request the participants to ask questions. Let me see if there are any questions, sir.
Sure.
Ashish, can you move Mr. Sabarano Baral to the panelist? You can also type in the chat box if you have any questions. There is one question, sir. Is it possible to apply this knowledge to Ayurvedic drug discovery?
I have no idea about the Ayurvedic drug discovery, but I believe you can try these molecules, but frankly speaking, I'm not from that area actually.
Okay. Any other question for sir? Sabarano, you can type in the chat box if it is possible for you. I think, sir, the session was really nice, and it was very clear to participants. I strongly believe that they have learned a lot of things from this session. So I would like to thank you for your time and for your efforts, and I really appreciate, sir. Whenever I have asked, you have always been ready to spare some time for these sessions. Thank you so much, sir.
Okay, thank you, brother. Thanks.
Bye, sir. So we will be closing then for today. So we have completed 3 sessions today. And tomorrow again, we will be starting. Session 1 for tomorrow is on genetic association studies by Professor Suresh Kumar Sharma from Punjab University, Chandigarh. And then we will have session 2 tomorrow, that is basics of deep learning by Alisha Praveen from Rostock University, Germany. Then tomorrow session 3, we will have we will have talk on microbial genomics by Dr. Avinash Sharma, who is a scientist at NCCS Pune. So I request all the participants to join in time tomorrow. And if you have any queries, you may drop in the WhatsApp group that we have created. I hope you are able to join the WhatsApp group where we are putting all the latest updates. If there are any updates, you will come to know about there. All right, and we will be sharing the feedback link. Yes, we will be sharing that soon. Yeah. Then you can, all the participants can please fill that form as well. And we are notifying on the attendance using the recordings from the sessions, as I informed earlier. Thank you, and let's see each other tomorrow. Have a good day. Bye.